Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article, a Phase 1-2 study of the antibody-drug conjugate Glembatumumab Vedotin, CDX-011 or CR011-vcMMAE, in patients with locally advanced or metastatic breast cancer by Joanna C. Bendel et al. This is the first study to investigate this antibody-drug conjugate in breast cancer patients and provides early evidence of clinical activity with an acceptable toxicity profile. My name is Roisin Connolly, and I am an Assistant Professor of Oncology at the Sidney Kimmel Comprehensive Cancer Center at the Johns Hopkins School of Medicine in Baltimore, Maryland, United States of America. My oncologic specialty is breast cancer and early drug development. There has been a surge in interest in the development of antibody-drug conjugates for the treatment of both hematologic and solid tumor malignancies in recent years. This class of therapeutic agents consists of a cytotoxic agent linked covalently to an antibody or antibody fragment, which is directed to a specific target expressed on the surface of cancer cells. A number of antibody-drug conjugates have been successful in clinical trials and have been approved for use in the clinic by the FDA. These include trastuzumab emtansine, or TDM1, for patients with HER2-positive advanced breast cancer, and brentuximab vedotin, which targets CD30 in hematologic malignancies. Glembatumumab vedotin, also known as CDX-011 or CR011-vcMMAE, comprises a fully human monoclonal antibody to a transmembrane glycoprotein, GPNMB, or glycoprotein non-metastatic B, which is covalently linked through a valine-citrulline linker to monomethyl auristatin E, referred to as MMAE in this podcast. MMAE is a highly potent dolastatin analog which targets tubulin. After binding to GPNMB expressed by breast cancer cells, the antibody-drug conjugate is internalized and MMAE is released into the cytoplasm through the action of lysosomal proteases, subsequently mediating cell cycle arrest and apoptosis via its microtubule inhibitory effects. GPNMB is an attractive target for the development of antibody-drug conjugate, as it is often expressed on the cell surface in high percentages of tumor cells and predominantly localizes to the intracellular compartments in normal cells, thus minimizing the risk of toxicity. It has been reported to be expressed in 40 to 60% of breast cancers. GPNMB appears to have multiple functions in malignant tumors, which include mediating intracellular adhesion, promoting tissue repair, regulating cell growth and differentiation, and down-modulating T-cell mediated responses, including anti-tumor immune responses. An early phase study in patients with advanced melanoma reported an overall response rate of 15% and an overall response rate of 28% in a subgroup of patients with strong GPNMB expression. Preclinical studies in breast cancer models indicated growth inhibition of GPNMB expressing cell lines and xenograft models when compared to control. Tumor epithelial GPNMB expression was also found to correlate with poor outcome in breast cancer patients and was an independent prognostic indicator of breast cancer recurrence. In triple-negative breast cancer, epithelial GPNMB expression was observed in 29.1% of breast cancer samples, correlated with shorter recurrence-free survival, and was an independent prognostic indicator of distant metastases in a multivariate model. These and other preclinical studies led to the investigation of Glembatumumab Vedotin in patients with advanced breast cancer. The article by Bendel et al., which is the focus of this JCO podcast, describes a phase 1/2 study designed to assess the hypothesis that Glembatumumab Vedotin will be safe and active in patients with locally advanced or metastatic breast cancer. The most common treatment-related toxicities included fatigue, rash, nausea, peripheral sensory neuropathy, and neutropenia, with few treatment-related serious adverse events. Dose reductions were required in 24% of patients treated at the recommended phase 2 dose of 1.88 mg per kilogram every 3 weeks. The primary efficacy endpoint for the phase 2 portion of the study was met, with progression-free survival at 12 weeks being 33% for 27 evaluable patients. GPNMB expression defined by greater than or equal to 5% of epithelial or stromal cells expression was analyzed in 15 patients treated at the phase 2 dose based on the availability of archival tissue. In 11 evaluable patients with GPNMB expression, progression-free survival at 12 weeks was 73% and overall response rate 18%. In 10 evaluable patients with triple-negative breast cancer, the progression-free survival at 12 weeks was 60% and overall response rate 20%, with 10% confirmed. Although this study is incorporated a relatively small number of patients with retrospective biomarker analysis, it has prompted further development of Glembatumumab Vedotin in GPNMB expressing breast cancer in the eMERGE and METRIC studies. Based on the paucity of effective therapies for patients with triple-negative breast cancer, this agent is of intense interest. The phase 2 eMERGE trial has been presented in abstract form thus far and enrolled 122 patients with heavily pretreated advanced GPNMB overexpressing breast cancer, as defined by GPNMB expression in greater than or equal to 5% of epithelial or stromal cells. Patients were randomized in a 2-to-1 fashion to Glembatumumab Vedotin versus investigator's choice of chemotherapy. The investigational agent was well tolerated with less hematologic toxicity than the control arm. The overall response rate for all patients was 19% in the Glembatumumab Vedotin arm versus 14% in the control arm. There was a statistically significant difference in overall response rate in the subgroup of patients with high GPNMB expression, as defined by greater than or equal to 25% tumor cell staining, 32% versus 13%, and also in median progression-free survival, 2.5 versus 1.4 months. In addition, in the patients with both triple-negative breast cancer and high GPNMB expression, there was a statistically significant difference in overall response rate, 36% versus 0%, and median progression-free survival, 3 versus 1.3 months. Final presentation of these data is eagerly awaited. These promising results have prompted the development of the phase 2 METRIC trial, in which 300 women with advanced GPNMB overexpressing triple-negative breast cancer will be randomized to Glembatumumab Vedotin versus capecitabine. This study is designed to support an application for accelerated approval of the antibody-drug conjugate in this patient population. If successful, Glembatumumab Vedotin will be the first targeted therapy to be approved for use in a subgroup of triple-negative breast cancers. Moving forward, it will be imperative to determine the optimal use of Glembatumumab Vedotin in breast cancer and other tumor types, as well as predictive biomarkers of response to the agent. Glembatumumab Vedotin does appear to target GPNMB in normal tissues, as evidenced by the development of a rash in approximately half of patients receiving treatment. However, the presence of a rash within the first cycle of therapy in the advanced melanoma trial appeared to be predictive of treatment response. Optimization of GPNMB expression as a biomarker will also be required. GPNMB expression was defined as greater than or equal to 5% of epithelial or stromal cells expression in the study by Bendel et al. And the eMERGE trial, however, the greatest benefit was observed in the eMERGE trial for patients with greater than or equal to 25% epithelial cell staining. As GPNMB expression is present in the stromal compartment in almost 70% of breast cancers, it will also be necessary to elucidate the contribution of these stromal cells in the setting of treatment with Glembatumumab Vedotin. Whether the use of archival samples to assess GPNMB expression is optimal is also unclear, as it is unknown whether expression levels change over time in tumor samples, vary between the primary or metastatic site, or if expression is maintained in specimens tested many years after collection. It has become clear that triple-negative breast cancer is a heterogeneous disease comprised of a number of different molecular subtypes. Glembatumumab Vedotin appears to be particularly promising in the GPNMB expressing subtype of triple-negative breast cancer. This agent may also warrant further investigation in non-triple-negative breast cancer subtypes and other malignancies. In 9 evaluable patients with HER2-positive breast cancer in the study by Bendel et al., the progression-free survival at 12 weeks was 30% and overall response rate 22%, with 11% confirmed. Finally, future efforts should consider combination strategies to enhance further the efficacy of this agent in carefully selected patient populations. In melanoma cell lines, tyrosine kinase inhibitors such as imatinib and ERK pathway inhibitors can enhance GPNMB expression on the cell surface and increase sensitivity to Glembatumumab Vedotin. In summary, this phase 1/2 study of Glembatumumab Vedotin in patients with locally advanced or metastatic breast cancer reveals promising clinical activity in GPNMB expressing breast cancers. If the phase 2 METRIC trial is a positive study, Glembatumumab Vedotin may be the first targeted therapy to be approved for use in advanced GPNMB expressing triple-negative breast cancer. This may herald an era in which triple-negative breast cancer itself is divided into many specific targetable subtypes based on molecular characteristics, with the promise of more individualized and effective therapies for breast cancer patients. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.